My name is Kate McKale, and I'm a biotechnology instructor at Norwich Technical High School. We have a biotechnology trade program here, which is a 3.5-year program where students get hands-on experience in laboratory training. In their junior year, our students get to show off their research skills and design their own experiment. They learn everything from experimental design, data analysis, communication, both presenting their research and communicating with mentors and industry partners. They learn documentation and maintaining a lab notebook for their research, and the art of technical writing, both making a poster, as you'll see, and presenting their work in an academic paper. Throughout our trade program and in this course, our students get a variety of hands-on skills just to get some background. We do plant biotechnology such as micropropagation. We do a lot of DNA extraction and quantification, so environmental DNA analysis. We do genetic engineering. We have microbial culture techniques such as gram staining, patch plating, biochemical assays for microbial cultures. We do bioinformatics and data analysis, additional biochemical assays, protein analysis, and then laboratory management, both how to run the lab and maintain all the laboratory equipment. My student today, Addison, will be introducing her project on sea urchins and sunscreen.
The title of my project is Impact of Sunscreen Contaminants on Sea Urchin Larva Growth and Development. My hypothesis for this study was that embryos exposed to reef-safe sunscreens would develop normally compared to those exposed to conventional sunscreens. This idea came from the growing concern over the chemicals found in sunscreens that wash into the ocean. Since 2008, scientists have linked these toxins to significant harm in marine ecosystems. In 2015, 15,425 parts per trillion of sunscreen were found in the water at Miami Beach. Research estimated that as much as 6,000 tons of sunscreen end up in the ocean each year, disrupting marine life. Some states and countries banned specific UV filters, including octocrylene, oxybenzone, and octinoxate, due to the harmful impact on the environment. To explore this further, I looked at how different sunscreens affected sea urchin larvae. Sea urchins are often used in toxicology research as they are highly sensitive to small amounts of pollution. I aim to find out if reef-safe sunscreens lived up to their label. To determine the concentrations of sunscreen, I used the data from Miami Beach. I used this concentration of 15,425 parts per trillion as the maximum concentration. Then dilutions were made to create 3 additional concentrations. To start, I injected the adult urchins with the 0.5 molar potassium chloride solution to stimulate gamete release. Once the eggs and sperm were collected, they were mixed for fertilization. After 2 days, the fertilized embryos were allowed to develop in 4 different concentrations of 10 different sunscreens. Observations were made for 27 days to monitor embryo development. The results showed that every sunscreen, whether labeled as conventional or reef-safe, caused abnormal development in the embryos. The sunscreen that caused the most abnormal development was Neutrogena. The sunscreen by Whole Foods showed the slowest rate of abnormal development, but it also contains one of the banned UV filters, octocrylene. The average date of embryo disintegration was day 5. The results led to an important conclusion. All UV filters, whether organic or inorganic, are toxic to sea urchins. Our study found that both types of filters, including those found in sunscreens marketed as reef safe or reef friendly, have harmful effects on sea urchin development. My research showed that there are no approved regulations on the terms reef safe, and they are just used for marketing purposes. Regardless of the label, the chemicals in all types of sunscreens disrupted normal growth and caused abnormal development in the embryos, showing that no sunscreen, regardless of its classification, is safe for marine organisms. My study had some limitations. Sunscreens contain multiple ingredients, and it's difficult to pinpoint which ones are exactly responsible. Another limitation was the stress placed on the sea urchins. The journey from Florida to Connecticut and the handling process could have weakened the urchins, affecting their immune systems and development.